What if a drug could do more than simply block a disease-causing protein? What if it could actually remove that protein from the cell? That is the idea behind PROTACs. And in 2026, this concept reached a major clinical milestone with the FDA approval of Vepdegestrant, the first FDA-approved heterobifunctional targeted protein degrader. So, how does a PROTAC actually make a cell eliminate a specific protein? Let's understand it step by step. Before understanding PROTACs, we need to understand one important cellular system, the Ubiquitin Proteasome system. Our cells constantly produce and remove proteins. When a protein is damaged, misfolded, or no longer needed, the cell can mark it for degradation. One of the important signals used for this process is ubiquitin. Ubiquitin is a small protein that can be attached to another protein as a kind of molecular degradation tag. But which enzyme decides that a particular protein should receive this tag? This is where E3 ubiquitin ligase come in. An E3 ubiquitin ligase is a protein complex that helps identify specific target proteins and facilitates their ubiquitination. In simple terms, think of an E3 ligase as a molecular matchmaker or tagging coordinator. It brings the target protein into position so that ubiquitin can be transferred onto it. Once the target protein receives the appropriate ubiquitin signal, the proteasome can recognize it and degrade it. Human cells have many different E3 ubiquitin ligases, and they recognize different proteins or operate in different cellular contexts. This diversity is extremely important for PROTAC technology because PROTACs can hijack this existing cellular machinery. Now let's see how. PROTAC stands for Proteolysis TArgeting Chimera. Unlike a conventional drug, which often works by binding to a protein and blocking its activity, a PROTAC is designed to bring a target protein together with an E3 ubiquitin ligase. A PROTAC is a bifunctional molecule. One end binds the target protein. The other end binds an E3 ubiquitin ligase. These two ends are connected by a chemical linker. So you can imagine the PROTAC as a molecular bridge, target binding ligand, linker, E3 ligase ligand. And this bridge is what initiates targeted protein degradation. First, the PROTAC encounters its target protein inside the cell. One end of the PROTAC specifically binds to that protein. For the clinically approved PROTAC Vepdegestrant, the target is the estrogen receptor, or ER. At this point, the target protein has simply been recognized. It has not yet been degraded. The next step is what makes degradation possible. Step 2. The other end of the PROTAC binds an E3 ubiquitin ligase. For Vepdegestrant, this involves the cereblon-containing E3 ubiquitin ligase machinery. Now the PROTAC has brought two components together, the target protein and the E3 ubiquitin ligase. The PROTAC therefore acts as a molecular bridge, creating a temporary target protein PROTAC E3 ligase complex. And this proximity allows the E3 ligase to facilitate ubiquitination of the target. In step 3, the E3 ubiquitin ligase helps add ubiquitin to the target protein. Additional ubiquitin molecules can be added, creating a signal that directs the target toward degradation. Remember, the PROTAC itself is not the molecular shredder. Its major role is to bring the target protein and the E3 ligase together so that the cell's existing ubiquitination machinery can mark the target for destruction. In step 4, the ubiquitinated target protein is then recognized by the 26S proteasome. The proteasome is one of the major protein degradation machines inside our cells. You can think of it as a highly organized molecular recycling and disposal system. It recognizes appropriately tagged proteins and processes them for degradation. Finally, the proteasome unfolds the target protein and breaks it down into smaller peptide fragments. And this is the key difference between a PROTAC and a conventional inhibitor. A conventional inhibitor generally works like this. The drug binds to the protein and inhibits its function, but importantly, the protein itself remains inside the cell. A PROTAC works differently. Unlike a conventional inhibitor, a PROTAC doesn't simply inhibit the protein. It physically removes the target protein from the cell. Now let's come to the major milestone. On May 1, 2026, the FDA-approved Vepdegestrant, marketed as Vepanu. It is an oral protein degrader targeting the estrogen receptor. The approval is for adults with estrogen receptor positive, HER2 negative, ESR1 mutated advanced or metastatic breast cancer whose disease has progressed after at least one line of endocrine therapy. Why target the estrogen receptor? In ER-positive breast cancer, estrogen receptor signaling can promote tumor cell growth. Endocrine therapies attempt to interfere with this signaling. However, resistance can develop. One important mechanism involves ESR1 mutations, which can contribute to resistance to endocrine therapy. Vepdegestrant takes a different approach. Instead of simply blocking the estrogen receptor, it is designed to recruit the receptor to the ubiquitin proteasome system and promote its degradation. The real excitement goes beyond one drug. Traditional drug discovery often asks, can we find a drug that binds to this protein and blocks it? PROTAC technology introduces another question. Can we recruit the cell's own machinery and eliminate this protein? This could potentially expand the range of proteins that can be targeted therapeutically. Researchers are investigating targeted protein degradation across areas including cancer, inflammation, neurological disorders, and other diseases. And PROTACs are only one approach. Another important strategy is molecular glue degraders, which promote interactions between target proteins and degradation machinery through a different mechanism. PROTACs are not without limitations. Their relatively large and complex structures can create challenges involving oral bioavailability, pharmacokinetics, tissue penetration, selectivity, safety, and the availability of suitable E3 ligases in different tissues. So targeted protein degradation is not a universal solution, but FDA approval of Vepdegestrant shows that this strategy can translate into clinical medicine. The future of pharmacology may therefore not only be about finding drugs that block proteins, it may increasingly involve designing drugs that tell the cell, find this protein and remove it. That is the promise of PROTACs and the emerging era of targeted protein degradation. So friends, if you found this video helpful, like the video, share it, and subscribe for more pharmacology and medical science content. Thanks for watching, and I'll see you in the next video.